So our last lecture, we ended with the microscopic structure of bone. That covered a lot of 7.2. In 7.3, we are going to examine bone growth and development. Basically, what we see is parts of the skeletal system begin to develop during the first few weeks of prenatal development. And overall, bones are going to form when bone tissue is going to replace the connective tissue along with that bone. This occurs in two ways. The two mechanisms involved with ossification includes intramembranous ossification and endochondral ossification. Remember that when you say ossify or use that prefix in general, it means bones. So you could call this intramembranous ossification or intramembranous bones. If we look at intramembranous bones, this type of ossification, the bones are going to originate within that sheet-like layers of connective tissue. They are broad and flat bones. So basically what we're looking at is flat bones, bones of the skull, clavicle, sternum, and some of the facial bones. Recall from microscopic structure of bone that the osteoblast are bone-forming cells. So when we look at intramembranous ossification, when you have basically connective tissue being filled in by bone tissue, that is a good example of where we see the infantile skull with the fontanelles closing up and hardening with bone growth there. And of course, the fontanelles of the infant skull would be what we would consider the soft spots. The other type of ossification is endochondral ossification. This begins as hyaline cartilage. It's going to form the model or the structure for future bones. And this is typically how most of the bones in the skeleton are formed. There are two basic types. If you look here at the diagram, you could see that endochondral ossification, most bones are going to form in this matter, and there are two basic processes. You have primary ossification and you have secondary ossification. So the hyaline cartilage is going to serve as the building model and going to allow for primary ossification to take place. Primary ossification in general is where you're going to have that center hardens as the fetus and infant develop. So what you see here is the center would develop in the hyaline cartilage tissue there. And then secondary ossification centers on the development in a child and then hardens during adolescence and early adulthood. So in this first picture here, which is highlighted in your textbook, you could see that primary ossification, you have the cartilaginous model, and then you have the developing periosteum, which would surround the bone, and then you have the calcified cartilage. And basically what will happen is in primary ossification, you get that where the center is going to harden. So right in here, you get that hardening center, and you get the development of compact bone formed by those, excuse me, formed by the osteoclast. Over here in secondary, or not osteo, yeah, over here in secondary ossification, which we see in adolescence and early adult, you are going to see the growth associated with this epiphyseal plate. And we'll look at that in a little bit there. So this would be long bone growth, typical long bone growth structure there. So during infancy and childhood, long bones lengthen entirely by growth at the epiphyseal plate. This is what we refer to as longitudinal growth. And basically, the bones grow in thickness by a process called appositional growth. So bones have both longitudinal growth and appositional growth. Appositional growth, uh, you probably know how to spell longitudinal. If I go back to the next slide here, let's click here. So growth is longitudinal and appositional. So let's basically look at growth at the epiphyseal plate. Here you can see the microscopic images. This is also described in your textbook, but we do have a couple zones. So if we look, the first zone is called the zone of resting cartilage. It is the first layer, and basically this is going to occur near the epiphysis, and you're going to have small scattered chondrocytes around this area that anchor to which anchor the plate itself to the epiphysis. So it's closest end of the epiphysis. These are resting cells, and it anchors that epiphyseal plate to the epiphysis. The next area would be the zone of proliferating cartilage. So this would be your second layer of cells. So here is that zone of resting cartilage. Here is the zone of proliferating cartilage, and look at the chondrocytes in there. And basically what you're going to have here is larger chondrocytes are going to appear, and they're going to resemble basically a stack of coins. And you can see that in the image over here. And ultimately what will happen is the chondrocytes are going to divide and replace those that have died at the diaphyseal surface of the epiphysis. So you have many rows of young cells that are continuing to undergo mitosis. In the next zone, you have the zone of hypertrophic cartilage. And basically what's going here is you're going to have extremely large chondrocytes that are arranged in columns. So the zone of hypertrophic cartilage, very large chondrocytes arranged in rows or columns. And basically what's going to happen here is these are going to be maturing chondrocytes. So you have maturing cells here. So older cells left behind when new cells appear, and the cells are enlarging basically as they are becoming calcified. And ultimately that would bring us to the next phase, or the next zone, which is the last zone, and that would be the zone of calcified cartilage. And in that zone of calcified cartilage, there is quite a bit that is going on. Ultimately, it's only a few cell layers thick. It consists of dead cells because the matrix around them has become calcified, so they no longer have nutrients feeding them. That calcified matrix is going to be destroyed by osteoclast. And basically, osteoclast, recall, are those large multinucleated cells that originate from white blood cells called monocytes. And those cells are responsible for bone resorption. Basically, what they do is they contain that cell organelle, the lysosomes. And if you remember from biology, lysosomes are kind of like the garbage disposal organelles of the cells. And what happens is they are going to secrete lysosomal enzymes that are going to digest that organic bone matrix. You also have acids that will be secreted, and the acid, of course, will be used to decompose the calcium salts into the ions Ca2 plus and PO4 minus. And then those ions can then go back into the blood, the calcium and phosphate ions. Remember, calcium ions, Ca2 plus and PO4 minus, are part of that hydroxyapatite. That's within that molecular formula there. So the matrix is then invaded by these bone building cells called osteoblast. And those bone building cells are going to lay down bone tissue on the calcified cartilage. And basically what happens then as a result, the diaphyseal border of the plate is firmly cemented to the bone of the diaphysis. So when we looked at bone structure, these are some of the things. So the diaphyseal border and the diaphysis is the shaft of the bone. And ultimately what this allows then is ultimately what this allows for then is the epiphyseal plate allows for bone lengthening into adulthood. As a child grows, what will happen is cartilage cells are produced by mitosis on the epiphyseal side of the plate, and then they are destroyed and replaced by the bone on the diaphyseal side of the plate. So what we see there is you have bone growth on one side, bone destruction on the other side, and therefore the thickness of the plate remains almost constant while the bone on the diaphyseal side increases in length. The cartilage of the epiphyseal plate is replaced by the bone forming that epiphyseal line. And if you look at your long bone structure, you will be able to see where they indicate that epiphyseal line. So ossification of most bones ultimately then is completed by age 25. To see this further, I think on page 209 of the textbook, you could see a timetable there, which illustrates, talks about some of the things that I've just discussed here. The next thing would be bone thickening. So we looked at longitudinal growth. How do bones thicken via appositional growth? It's not as in depth. Basically what we see is, along with increasing the length of bones, you get this thickness in the bone's diameter. And basically, this occurs in the osteogenic layer of that periosteum. So we know that long bones themselves, they have that area called the periosteum, and osteogenic. If you're looking at genic, that if you break down that word osteogenic, you're looking at forming, and osteos bone. So you're looking at bone formation here, and it's going to be growing outward. So osteoblast lay down matrix of compact bone on the outer surface, and basically this is accompanied by osteoclast destroying the bone matrix at the endosteal surface. So you have this appositional growth occurring all along that periosteum there. So that does take us through bone growth. Now let's look at development. So homeostasis of bone tissue. So basically, once bones are formed, the action of osteoclast and osteoblast is continually remodeling the bone. Osteoclasts are going to reabsorb bone. Osteoblast replace the bone. And these opposing processes are highly regulated, so the total mass of bone tissue in the adult skeleton is going to remain constant, even though 3 to 5% of bone calcium is exchanged each year. So there are some factors that we do see that affect bone growth and repair. A number of factors that influence bone development, growth, and repair of bone include nutrition, exposure to sunlight, hormonal secretions, and physical exercise. So if we look at the first one, nutrition, basically we have 3 vitamins, vitamins D, A, and C, and you can see generally what those vitamins do. But vitamin D greatly increases intestinal absorption of dietary calcium, and it's going to retard its urine loss. So basically what vitamin D, if we look at it, vitamin D deficiency causes rickets in children. It causes osteomalacia in adults. Vitamin D is found in dairy products, and it is synthesized in the skin when you have exposure to sunlight. On the other hand, you have vitamin A. Vitamin A is required for bone reabsorption controls. And basically what's going to happen there is it's going to control the activity and distribution and coordination of those osteoblasts and osteoclasts during development. So if you have a deficiency in vitamin A, of course you can see that generally it's going to retard the bone growth, and that's due to the activity, distribution, and coordination of those osteoblasts and osteoclasts. And then lastly, you have vitamin C, and vitamin C is going to help maintain bone matrix, which is collagen synthesis, and a deficiency in vitamin C, of course, would be scurvy. Then you have sunlight. Exposure to sunlight does have an impact on bone growth, development, and repair. You also have hormonal secretions. One of the hormones discussed is HGH, lowercase h, capital G, capital H, is the abbreviation for human growth hormone. And basically human growth hormone from the pituitary gland is going to be responsible for the general growth of all tissues. In bone, basically what the human growth hormone does is it causes hyposecretion in childhood, which results in pituitary dwarfism. If you have hypersecretion in childhood, it results in gigantism. So hyposecretion, you would see basically little people, and then hypersecretion. And look at those two prefixes. Hypo means below, so you're going below the average secretion levels, and hyper is you're going above the average secretion levels of HGH. Hypersecretion in adulthood results in acromegaly. These disorders are discussed in greater detail within the chapter 13 when we get there. Lastly, what we do see is the other hormone, which is the thyroid hormone. Thyroid hormone, there are two of them. You have T4, which is called thyroxine, THYROXINE. And thyroxine is going to be that thyroid hormone that is involved in the replacement of cartilage by bone in that epiphyseal plate. The other one is calcitonin, and that's going to act on several sites to decrease blood calcium levels. So calcitonin, the correct spelling of that would be CALCITONIN. You also have parathyroid hormone, PTH, all capital letters for the abbreviation there. Parathyroid hormone, basically the gland is going to stimulate osteoclast to reabsorb bone. And then the last group of hormones are the sex hormones from the male and female gonads, testosterone for the male respectively, and estrogen for the female respectively. Basically, the sex hormones are abundant at puberty and cause considerable long bone growth. And ultimately what this does is it aids osteoblast activity and promotes new bone growth. It also degenerates cartilage cells along the epiphyseal plate. Therefore, the epiphyseal plate is going to close, and estrogen effect is greater than the androgen effect. Lastly, physical exercise. When you have stress, it does stimulate bone growth, hypertrophy. Lack of exercise leads to bone tissue wasting away, and that would be atrophy. Atrophy is when you start to break down that bone tissue because of lack of exercise. I would look over. There is a clinical application on 7.1 which discusses fractures. I think it's around 212. Let me look. Yes, on page 212 of the textbook. Be sure to read over the clinical application dealing with fractures. So our last section that I think we have to go over is basically a little bit on bone function, and that will end our discussion on the physiology of bone as we continue to dissect the skeleton, both the axial and appendicular skeleton, for the lab practical. So the lecture test will include some basic anatomy, but also hit more heavily emphasizing the physiology parts of the skeletal system. That's it for this lecture. We will continue with bone functioning in our last lecture for this chapter. Have a great day, everybody.